Hello again, everyone.
Yeah, okay. So, hello,
Hello.
hello.
Hello, everyone.
Yeah, maybe we might wait 1 minute. Yeah. Okay, I think that maybe we start because we don't have much time. So, yeah. So section four, we happy to invite Juan Pablo to make some introduction of the the EBI biodiversity portals, and this global biodiversity portal is a centralized open access database, and that integrates different or store different genomics information collected from the genome projects, from the Earth biogenome projects. And this portal offer some user-friendly search filters and some other fictions. I think Dr. Juan Pablo will make more detailed introduction about.
Yeah, hello? Okay, can you hear me, right?
We can.
Yeah, I can hear
Perfect.
you.
Perfect. I'm going to share my screen.
Okay.
Yes, are you seeing my slides?
Yeah, we can see that.
And now?
Yes.
Okay, perfect. Thank you very much. So, good morning, afternoon, and evening, everyone. My name is Juan Pablo. I am an ARISE Fellow at the European Bioinformatics Institute, working with the Agricultural and Biodiversity Coordination team. To maintain and improve the biodiversity portals at EMBL EBI, I have recently started as an ARISE Fellow. So today I am speaking on behalf of my team, and I will show you what they have developed so far with the biodiversity portal. I'll also tell you a little bit about what are my plans with this portal. So, yep. As many of you may already know, the Air Biogenome Project is a global initiative with the goal of sequencing and producing high-quality chromosome-level reference genomes for all eukaryotic species on Earth. It serves as an umbrella for various sub-projects, and it encompasses efforts that focus on specific regions, species groups, and ecologically significant organisms. The pace of genomes produced is higher than ever. The last time I checked, the Darwin Tree of Life had produced around 1,200 genomes only, but I'm sure that the number may have increased by hundreds right now if we were to check it today or to look at any of the other projects that are working under this global initiative. This means that the amount of genomic data and metadata available is increasing fast. So having data services that facilitate access and provide basic insights to these genomes is essential. The Biodiversity Portal is a comprehensive data service providing access to reference genomes from a diverse range of the species generated by the EBP, and it centralizes the access to sample metadata, genomic assemblies, and annotations that are hosted by EMBL EBI resources such as ENA, Ensemble, and Biosamples. And it is funded by the EMBL Planetary Biology theme. Along this presentation, you will see sometimes some QR codes that you can scan to go to the different sections I will be referring to. The website is structured into 4 main sections. We have a data portal offering access to genome data and metadata. We have a tracking system for monitoring the species sequencing progress across different biodiversity projects, a sampling map that visualizes the geolocation of collected sample of the species, and a project page which links to the dedicated websites of various biodiversity sequencing initiatives. So now let's have a quick tour for each one of these sections. The portal features a simple table-like design with a user-friendly search bar. You can search by scientific name, genus, species, or both, or also common name to find the genome metadata of your species of interest. If you, for example, receive a very long list of results, you can also refine that search by using filters for the genome sequencing status, specific projects, or even the phylogenetic rank. Additionally, you can also try to search for genomes that are associated to symbions or metagenomes. Users can customize the metadata that is displayed on the portal for depending on their information needs, and they can download this metadata as a CSV file for future reference. The search results also include links to external resources such as GOAT, as well as the associated publications for different exploration of genome-related data. Once you have found the species you are looking for, click it on its scientific name. Will take you to the species detail page. And this page contains the scientific name, common name, and the tree of life identifiers. That page is organized into 4 tabs. The metadata tab provides detailed species and sample metadata, along with the links to more comprehensive information available on biosamples. The tables tab includes 3 searchable tables, one with annotations containing the links to download the data available, and also with links directed to the EMBL genome browser if you want to explore more in detail the annotations. Also, you have related assemblies table that contains the links to all the assembly data in the ENA service, and a related files table that contains relevant raw data like CRAN files, BAM files, and so on. We also have a genome tab, genome node tab, which contains a link to the scientific publications that is reporting that reference genome. And the GOAT tab, the Genomes on a Tree service, this tab displays some of the metadata at GOAT and provide a link to further explore the metadata available at the service. And we are planning to kind of deepen that collaboration with GOAT to bring more data and be able to visualize the data here. The sampling map section displays the geolocation of collected samples and species. You can search for the specific species using the search bar and or applying different filters like sequencing the status, projects, and also the phylogenetic groups. The map allows you to zoom in to view the species and samples at specific geographic locations. And by clicking on the species name, you'll be redirected to the species detail page information for further exploration. Right now, we are developing the sampling map portal, so our users can retrieve all the samples in a specific geographic location by clicking on any bubble in the map. You also, using Google Street View, you could look at the approximate location where the samples were collected, which I think is a nice feature. And this is in development right now in the main biodiversity portal, but the QR code will take you to how it look like in the detailed data portal, which has this feature fully developed. The tracking system section enables users to monitor the sequencing progress of a species through each stage of the sequencing pipelines, from submission to biosamples to the final submission of the annotation files to the ENA service archive. You can search for your species of interest using also the search bar and apply the same filters that I have mentioned before. This biodiversity portal centralizes data from various projects under the Earth Biogenome project and provide access to each one of the individual dedicated websites if you want to explore that information further. For people that prefers programmatic access over the user interface, we have an API access to the genome's metadata. You can use the species name to get all the metadata associated in our portal, and also all the URLs associated to the files in the ENA service. Also, we have a downloadable Python app aiming to facilitate data downloads. This Python app can be downloaded from GitHub or clone, and you can run the script in the command line or use a Docker image to download all the data that you want. All the instructions are on GitHub if you want to look in more detail. With it, you can use different parameters to find the data metadata you are looking for. So you narrow down by project names, species data status, click, or taxon group, and you can also use a list of species to get the data that you want. Once you are installed that app, you can specify the parameters in the command line to begin downloading your data sets. As in this example, I am showing how to download the annotation data sets for the species Apamea sortens. And then in your computer, in your downloaded location, you will have a folder with all the related data sets, the GGF3, the GTF, and so on. So this was a very quick overview of the portal's features. And now let's take a look of our plans for any new functionalities. This is related mainly to the fellowship I proposed to the ARISE program. So currently, the portal works well if you know the species you are searching for, providing you with a quick access to the data you have an interest in. However, we would like to go a little bit further and enable a little bit more of or deeper exploration of the data and associated metadata in the portal interface. So we want to do this because it's important to recognize that there is a strong interest within the scientific community in integrating data across the biological sciences. So we have many data infrastructures that are maintained by bioinformaticians and from the side of biodiversity informatics that are keeping valuable information about almost all level of biological organizations from molecules to ecosystems. And integrating this molecular data with biodiversity data, such as the genomics and the ecological information, has been promoted as a crucial step for enabling integrative analysis in biology. And even some say that for addressing global challenges like the biodiversity monitoring tasks or assessing the impacts of climate change. By sequencing the genomes of all eukaryotic species, the Earth Biogenome Project is deepening the use of this molecular data by users from areas outside molecular biology, like systematic biologists, ecologists, and among others. And we need to think in ways of browsing this data, exploring it, and presenting it in a way that aligns with what these communities of users need and the way they normally portray their data in their analysis. So, for example, annotation data plays a key role in comparative biology in general. Systematic biologists use data like genome size or specific genes to characterize and classify species in their taxonomic treatments. So presenting kind of broad summaries of genome counts, features per taxonomic groups may be of interest to them. Evolutionary biologists are interested in exploring the evolution of genome features in phylogenetic contexts. So providing these genome counts and features in a comparative way using a phylogenetic backbone is always a must for these portals. And ecologists and evolutionary biologists alike are interested in how genomic features such as genome size may vary with species distribution and how they correlate with known biogeographic patterns like latitudinal diversity patterns or with climatic variables such as temperature and precipitation. Therefore, is like being able to browse the genomic data that we have in our portals by using geographic location of a species, or maybe a landscape classification showing habitats, ecoregions, and so on. And even environmental regions may allow some scientists to narrow down the data sets that they need for this specific analysis. So to achieve this goal, we are focusing in two key improvements for our portal. First, we want to expand the metadata available to include genome dataset quality metrics and annotation statistics, metadata about the biological context, such as the species geographic range or distribution from using GWF data, and by using this, linking to environmental data like climate or other biogeographical regions, doing spatial classification analysis. And ultimately, this will allow us to filter in this dataset by that geographic distribution and environmental ranges. Second, we also want to improve data visualization with interactive dashboards that try to display these patterns of genome feature variation in relation to geographic location and climatic factors, and enable the comparison of genomic features across a species using a phylogenetic framework. Sorry. Yeah, and given the variety of data types and very sparse data sources around, we plan to implement a data warehouse to centralize some snapshots of this information. This will facilitate the data exploration by enabling users to query and visualize everything from one place, while still providing some links to the original data sets in their respective data services. We are currently exploring the feasibility of using Google BigQuery as our cloud-based data warehouse solution to integrate annotation data with GW's distribution data. And this is because we are taking advantage that GVIF distribution data is already in the cloud, and maybe some there are also some climate data products that are out there that we could use and try to integrate all this data together in one place. Our portals are also deployed using Google Cloud products, so it seems like natural to us to first explore this possibility. And we are building all the data pipelines using Python Apache Beam and trying to orchestrate our workflows using Apache Airflow. So yeah, so right now, yes, we are in the process of building these pipelines, and yeah, and we will show you soon some of those results. So we plan to have like a prototype of our first dashboard ready by early next year. And from that point, start to show what we have to the community in order to kind of have a user-driven research cycle. The idea is having first a dashboard that is able to display how the annotation and genomic features changes in geographical space, while using different landscape classification like biogeographic regions, biomes, and so on. This is the team that is behind the development of the portal. And we would like to hear from you. If you are already one of our users, please feel free to contact us at biodiversityportal@ebi@ac.uk. And if you are already a user, please go and fill our user experience, because we are really keen to improve the services that we already have. At last, thank you very much to all of you for listening to me today, for the invitation to share what we are doing and what our plans. I would like to thank the ARISE program, which is the career accelerator for research infrastructures in EMBO, who is paying for my fellowship, and also the European Union's Horizon 2020 Research and Innovation Program. And of course, to all the Biogenomics project people associated, because this wouldn't be possible without you. And I am sure that what we are doing together is going to open new ways of asking questions in biology in general, and to the planetary biology team for funding our portal. I will be happy to listen to you, your questions, comments, and thank you so much for listening again. Thank you.
Yeah, thank you. I thought Emilio had questions. Yeah, maybe we can keep these questions because I also have questions. So I think we need to move to the next presentations, because we will keep it at the end of these sections. And we are also happy to invite Dr. Zheng Li, Dr. Li from the Ghent University, and it's from the Ghent University, the Department of the Plant Biotechnology and Bioinformatics. And Dr. Li is an assistant professor at Ghent University, and his research employed comparative genomics and hyprogenomics to unravel the complexity of the plant genome evolution and also the context of the ancient prioritizations and their roles in plant adaptations and diversifications. So welcome, Dr. Li. Yeah, welcome. You can
hello.
hear me?
Yeah, I can hear you well. Can you hear me all?
Okay.
Yeah. Good,
yeah,
perfect.
okay.
Share my slides first. Can you see my slides now?
Yeah.
Yeah, okay, cool. Yeah, so, well, thank you for the introduction. So I'm actually mainly the downstream users of all the genome projects actually. So so today, well, mainly I would like to discuss let's say an issue that we met when we were trying to identify whole genome duplication events using like genomes, and then we found that so wow, for the following genomic analysis, it's not really just a simple scale up of all the analysis, but there are actually some other issues that would actually pop up when we have more and more genomic data. So just a simple introduction for people who are probably not really familiar with polyploidization. So polyploidy is the condition of having more than 2 sets of chromosomes or haplotyped chromosomes. And around us, there are so many polyploid species. If we go to a supermarket, I mean, or if we're in the wild and we found a population of species of plants, and there are quite some individuals, they are actually polyploids. And we have wheat, carps, and also amphibians. There are species that there are polyploids in those species. And also for Asian polyploids, it's actually refers to polyploidization, or sometimes we see we call them whole genome duplication events that occurred actually in the very distant past. So in 1970, Susanna Ono wrote a book about gene duplication, so titled Evolution by Gene Duplication, where he only looked at very limited genomic data, well, I mean, molecular data, so much fewer than what we have at the moment. And he proposed that whole genome duplication have occurred at least twice, actually, during the evolution history of vertebrates. And after 50 years, so we sequenced a lot of genomes nowadays, and in the end, we could really verify his hypothesis that there are two whole genome duplication events actually occurred before the divergence of vertebrates. And also for green plants, because I studied mainly focused on plants, so there are many species that they are Asian polyploids. And here is actually a phylogenetic tree that we built in 2022, where we used all the published plant genomes, and we mapped the whole genome duplication events on the tree, and those rectangulars on the trees are the identified whole genome duplication events using the data before. That's quite a lot already, and it was actually quite challenging already. And I could say we would actually have more challenges when we have all the genomes nowadays. Oh, they're on the way. So to identify whole genome duplications, we use like three approaches mainly at the moment. One is collinearity or synteny. So it's actually a way to look for the genomic structure of duplicated genes. And we also use an approach called KSH distribution, which actually we try to measure the divergence time between duplicated genes and to see when whole genome duplication have occurred. And the last one we call is gene tree species tree reconciliation, and where we actually try to look for where on the species tree a duplication event actually creates the duplicated genes. So those are the approaches that we use. And then we actually noticed that there are some issues when there are more data. So when the data increase, we could see some issues, especially when in 2019, when the 1,000 plant genome, 1,000 plant transcriptome data was published, where we found actually, they identified a lot of whole genome duplication events, and then we found, okay, then they found a lot. So those red branches on this tree, they are the whole genome duplication events. And then we think, okay, because before we met some issues, maybe, or do they have the same issue or not? So I will first introduce the issue that we met before. Oops. Yeah. So it's here. So previously, when we have very sparse taxon sampling, so we always we use like one lineage to represent a family or something. So like for orchids, so that's the first orchid genome that we worked on. And in this project, we actually found for Phalaenopsis. So we use those Phalaenopsis to represent the whole family of orchids back then. And where we actually identified a whole genome duplication events at 76 million years ago. And then for the second one, it's okay. So it's Dendrobium, and the species actually diverged from Phalaenopsis at 39 million years ago. And that's safe actually because for the whole genome duplication event, we found it's actually have occurred 76 million years ago, which is actually quite earlier than the divergence between those two species. So those two species, they shared the same whole genome duplication events. But when we were actually working on the third orchid genome, so the Apostasia genome, which is actually the earliest diverged lineage of orchids, we found that the divergence time for the crown age of orchids actually quite, the period, the estimates are quite different in different studies. And here I cited 4 studies. It shows a very broad range for the crown age of orchids family. So from 100 million years to 63 million years. And then when we put the whole genome duplication events that we identified in Phalaenopsis, then we found, okay, then it falls actually within the crown age estimate And then we are actually quite not sure whether then the whole genome duplication events has occurred before the divergence of all the orchids or it's actually afterwards. So then we actually we worked a lot on it and see what's happening behind that. And the one thing we noticed that the like different lineages, they could have different substitution rates. So in this case, I gave a very simple example. So for two species A and B, one A has actually a higher substitution rate, and they both share a whole genome duplication event in this case. And then for the KSH distribution, so where we look at the time divergence of duplicated genes, and if there's a whole genome duplication event, then we would actually expect to see a peak actually in those KSH distribution. So for the purple one, so the KS distribution from species A, it actually has a higher KS value than the yellow species, so species B. And then of course we want to place the whole genome duplication events actually on a species tree. So we here put Oslo KS distribution between species A and B. And then we can see, so here I use a rectangular to show the peak of the Oslo KS distribution. And we can see that actually for the purple species, the whole genome duplication event has actually occurred before the speciation event, while for the yellow one, it's actually after, right? So that's the results that we could have. And we actually end up with two whole genome duplication events actually on this lineage, which is not really true here. And there's a hidden assumption that, okay, the A and B, they have the same substitution rates, but they are not. So that's one issue, which has been actually ignored by previous studies, actually when we scale up the analysis. And the second approach is, the second issue is some uncertainties in gene tree and species tree reconciliation. So when we look for gene trees to describe their evolution, in most cases, we only end up with one gene tree. So we use like maximum likelihood tree or a consensus tree from Bayesian approaches. And we are not sure whether the gene tree is really correct or not. And if it's an incorrect gene tree, then when we perform the gene tree species tree reconciliation, then we tend to put the duplication event towards to the root, and then the gene loss event towards to the tips. And also for the uncertainties here, of course, we have bootstrapping value or posterior probability, like kind of mirror of the uncertainties. But then when we want to remove the uncertain subtrees, we use arbitrary cutoff, like 50%, for instance, of the bootstrapping value or 70. And we don't know which one is good, actually. And the third thing is actually for the uncertainties in reconciliation. So for gene tree species tree reconciliation, we try to put evolutionary events on the species tree and try to explain the evolution of the gene tree. And when we put the when we put evolutionary event, we have no measure yet, like how confident we are we could put event on that on a specific branch of the tree. So that's also another uncertainties. And the fourth one is actually for this approach, we are actually not sure which the type of duplications that creates the paradox. So it could be from small scale duplications. So then on the recent branch, so on the branch leading to tips, then they would actually have more duplication events. And also on very long branch, we would also expect to see more duplication events. And then when we call whole genome duplication, we also always say, okay, if at the nodes, they're like 100 or 200 or even sometimes maybe 500 duplication events, then we call there's a whole genome duplication event there. But again, there's no real benchmark or benchwork to test like how many what the cutoff would be actually, and it really depends on the data sets. So based on all those uncertainties, we would end up sometimes with a case that whole genome duplications could be everywhere. So that's actually kind of the thing for the 1KP project. And so we really want to propose a workflow that could actually kind of solve the issue. But of course, again, for scale up our analysis, I think we still have some issues. So here I just show a little bit result from a published paper actually. So we actually look at the 1KP data with some data from ferns. So here we only use 16 species. So from the 1KP paper, they actually identified 10 whole genome duplication events in the lineage of fern, the leptospongiate ferns. And then interestingly, actually 1 year later, there's another group. They published a very similar data set with more or less the same species. They also identified 10 whole genome duplication events. And then we were also quite interested in then whether those two results they agree with each other. And to our surprise, actually only 5 of those events are identical. And then further, we consider about the substitution rates and also the uncertainties for gene tree species tree reconciliation. Then we found, okay, for the 1KP data set, wow, we could only actually verify the 4 whole genome duplication events. And then for the paper from Huang, I think we could only verify 2 whole genome duplication events from there. And we also noticed that they missed 2 whole genome duplication events. So now we are actually trying to develop a R package. I'm not sure it's a good solution to solve the problem, but we try to put those kind of things. And yeah, I hope at least we could analyze 20 or 30 species in one run. So it's an R package, and which can be actually installed on other everyone's laptop. So which actually puts like the CDS and the GFF file in all one location, and then it can produce like a script, and then everyone can run the script locally, if like for bioinformatician, and then they can upload the results to the R package again with a graphic user interface, and then can do all the analysis based on the results. That's kind of our current solution. I know it's not ideal. So we are also trying to develop a web server where the users could upload their data. Maybe then we could actually also do some analysis. But we also have some issues like to find computational resources actually to really implement the service. So here's a little one. It's still under developing, and that's kind of analysis for the KS. It's a little demo. So when the user upload the data, then they can select the species that they want. And then, of course, then it can just perform the, for example, to draw a KS distribution here. It's a little bit slow at the moment because we also did some other analysis, like for the Gaussian mixture modeling and the sizer analysis. And then, of course, users can adjust the figure somehow. So that's a little case. And then also they could play with some parameters like the weights and the bin width. Yeah. And then, of course, then they can later download the figure, and then it's a figure could be used in publication, we think. Yeah, so here then the user could actually also perform the rate correction. So here is a way to perform, one way to perform actually the rate corrections here. But yeah, our principle is actually, okay, we produce the scripts and then the user could run the scripts locally, because it's still kind of heavy computation actually. Somehow it could take some time to finally get the results. Okay, this part is done. And then we hope when the user gets their results, then everything is in the same folder, and then they can upload the folder again, either locally or remotely, and then perform their further analysis. And that's actually just a way to map all the whole application events on a phylogenetic tree. Yeah, and then of course user could actually adjust or modify the whole application events and perform some artwork. Yeah, okay, all right. That's actually my talk. Thank you very much.
Thank you, Dr. Lee. I know you are still business chief in Taipei.
Yeah, I'm in Taipei now. Yeah.
Yes, thank you so much. Yeah.
Yeah, it's
So
really my pleasure. Yeah.
yeah, because I speak the Minnan dialect. Yeah,
Oh, really?
yeah.
Oh,
So
cool.
yeah, but I've never been to Taipei before.
Okay.
Yeah,
Yeah.
yeah,
It's really nice here.
yes.
Yeah.
Oh, okay. Yeah, I hope that one day I can go there.
Yeah.
Okay. So thank you, Dr. Lee again. So
Yeah. Thank you.
yeah. So the last presentation is from the Jerry and from National Gene Bank, and now this system is still under development, but you will show some concept and make some brief introduction about this part. So welcome, Jerry.
Hello, everyone hear me?
Yeah,
Okay, I
you can
will
just share your
show
screen. Yeah.
my script.
Okay.
Okay. Can everyone see my slide? Okay. So thank you for the invitations to share some of our latest work here. So since the OpenAI ChatGPT took off last year, so we have been contemplating how to get involved in the AI or something else. And we've considered developing large language models for the biologies and also think about to constructing the data sets required for AI trainings, regardless of the approach. So as a database, to meeting the user's needs has always been paramount. So therefore, we've developed the biomical tools now. So the many friends in these sessions are likely familiar with the National Gene Bank information, the bioinformatics database. So we primarily have the CNSAs, a data submission system, which has currently collected approximately 60 petabytes of data from research users. And in addition, we also have 3 major portals, including the genome portals, which mainly collect the genome assemblies and also its annotations, and the single cell portals and special temporal omic portals. And our special temporal omic portals was also just published in the NAR journals last year. So with so much data, in addition to curating data to enhance its quality and usability, we need to provide the most convenient way for the users to access the data they desire. So therefore, for those who are new to bioinformatics analysis or want to quickly access the required data, being able to obtain all the needed data through a single entry point is a very cool thing. So to achieve this goals, we have developed the Biomical online tool, which is based on generative artificial intelligence, and it will understanding the user's nature, semantics, and help users find the data they needed. So it can even carry out some simple analysis as well as the visualization task. So to achieve the above purpose, so this analysis platforms should meet such as a framework. Like the general GPT type tools, it can be divided into 5 steps. So the firstly, the use ask questions. The AI will first determine the user's intent for classification, such as whether their goal is data searching or to do the analysis or visualization. So since the AI models we use general purpose, so understanding the biological significance is not so easy. So it's necessary to limit the user's questions based on the prompt. And then the second step after the AI recognize the intent is to catalog the user's needs for the next step of judgment. intents are divided into the data searchings and also the analysis and visualization. And the third step after the AI classify the user's intents is to proceed with parsings. If it's the search part, the first browser the metadata of the data sets in the database to determine the required data sets, and then enter the data set to identify the needed data engines. Finally, the Biomigo will feedback the information corresponding to that engines to the users. So if it's the analysis or visualization models, it will go through the existing models, identify the most needs for the users, and call it out on the main page. And to the first step, it will confirm the user can confirm the results and proceed to the next operations. And this step accurately no longer require the participations of artificial intelligence in the search scenarios. If it is confirmed as the needed data, the users can confirm the operations, and the system return the detailed information of the data and also the download link of the data. But in the analysis scenarios, if it is confirmed as the needed analysis models, the users can upload the files and do the input and adjust the analysis parameters, and then start running, waiting for a moment, and it will get the results. And the final step, the user can download the search data or the analysis or visualization results, and can also add the result to the built-in favorites of the Biomigos for unify the management. So this is the main interface of Biomigos, similar to common AI tools. Of course, we have removed its chat function, so you can only carry out some research work on it. So this is the dialog box where all search informations and the model information will be feedback here. And below is the input box where the user type in their own needs. So the AI can judge the user's needs based on natural semantics. And on the right side is prompt informations, and we will prepare in advance the sentence patterns for searching and analyzing model calls, so that the user's understanding what kind of the questions methods is the easiest to get the desired result. So on the left is the dialog box management. The users can open multiple dialog boxes to manage different projects. And the right side is in this part is the favorites. Some searching results or analysis results can be also collected from the next used. And we also provide some help documents as well as the user's information editing features. Okay, so for the data search scenarios, the users first enter the demand information, and the AI will search and feedback the following tables. For the genomic projects, the table will feedback or matching the genomics in our database, and with each rows corresponding to an assembly. After confirming the needed data, it can be collected so that the data can be put into the right favorites, so which is convenient for the future use. And you can also directly click to view the detailed information and download the data directly without the collecting. And this is the scenarios of the analysis modules. We have the modules such as the transfer the CDS to proteins, or do the alignments or the phylogenetic trees, and also plot the case distribution, something like that. And after the users type in the demand, the AI match the analysis modules and call it out. And the analysis module consists of several parts. The first, there will be an introduction to the modules. Below, there will be the parameter information that needed to be typed in. and marked as required or optionals. After uploading the analysis input files, it can be run. And if you are not sure about the format of the analysis model input file, you can also view the prepare example files. So, and then click the run and wait for a while, and the analysis result will be displayed below. And user can also collect the analysis result to the lab varieties or directly view and download. And the current page also provide the data browsing. So if you are not satisfied with the analysis result, you can directly adjust the analysis parameters and click run again to wait for a moment and got the next result from the run. And the visualization model is also designed. Firstly, according to the user's visualization needs, it will the AI will match the visualization models we put in the management system, and there will be introduction informations for the models and parameters needed for drawings, and which can be adjusted according to the specific needs. And after running, the visualization results can be also directly opened for browsings and downloading, and can also be collected to the lab varieties for the next use. So for the Biomigo, the front end need to feedback data sets and analysis models based on the users needed. So the background management system need to be different from the previously database. Therefore, we have also developed a background management platform to smoothly add the data sets, the analysis, and the visualization models. And our background system is divided into 5 major systems, including the dataset, the prompt, the modular management, the model management, and also the user statistic centers. The dataset management system mainly converts external data resources into the format that the AI can easily read. We organize the data of the 3 portals from the 3 portal of CNGPDB into the table forms and add them as different data resources and retain the metadata that the AI can easily understand, such as the source specials of the data. And there will also be more details information table corresponding to metadata for users reading. And that is the prompt management, and modular management are also convenient for the administrators to batch add in the backgrounds or import it in the JSON format. And the information of single models include the model's introductions, and which is mainly for the AI recognitions, and as well as the definition of the input and output parameters, and the corresponding script and the other results. So the software and the script involved in the analysis will be also deployed on our service in advance. So in addition, there is the model management system. Okay. So this is the model management system where we can change the core intent recognition models according to the performance of different. So currently we support the models like the Moonshoot or the ChatGPT from OpenAI, and also the GLM. And the user centers mainly detect users' usage habit to provide better services. Okay, so this is the specific situation of our background management modules. In the dataset management department, the administrators can upload metadata at any time in addition to genomic and the transcriptome data, and also the literature information, the database information, and also the knowledge text can be uploaded in the table forms to expand the function of the biomigos, because many of the metadata can be also recognized by the AI, right? So the module management part can carry out the batch management of the modules. So it can be import and export the model information in JSON format and adjust the activations status of the models. The models that are not activated will not be matched by AI. So in addition to the JSON format, the model information can also be added in the form of online tables. Therefore, many external users can also help us to maintain the Biomigo tool together. So in the models management part, there is also a windows for general questions and answer interactions. So it will provide it for the administrators to carry out the basic debug. And then the user data center will feedback the usage of the users, especially the core situations of data sets, the analysis, and also the visualization models, which is convenient for us to understanding which data sets and model are the most popular. Okay, so the last slide to sum up, as a database, a large amount of data has been collected. So in order to make the users better reach this data, there is the artificial intelligence-based tools Biomigo. So the essence is to facilitate the users to quickly obtain the required data on the same page so that the data can flow, right? So in addition, the users need to carry out some routine analysis and visualization when they get the data. So since the data search is used, we will try to provide a simple and easy to use online analysis tool so that the users can carry out some research work in a short time without the background of the software's configuration and also the code foundations and the collect, and it will help them in an efficiency ways to collect the result for the users. And finally, the users can also provide us with some script and tools they often use, which can help more scientific researchers, like the Professor Lee Zhen, who just made a report. He have also discussed whether to add tools and corresponding database data sets for the whole genome duplication analyzed to Biomigo. I think it's will be a nice try so that everyone has more time to think about their own scientific research topic rather than spend more time on how to do the configuration for a bioinformatics tools. Okay, so that's all for my report. So thank you, everyone. And the Biomigo will offshore launch in the next month. Now is in the development now. Okay, thank you.
Okay, thank you, Jerry. So I found that there is 2 questions, but I'm not sure that the Matthew and Emilio already get your answer.
Yes, yes.
Okay. So I think I need to close this workshop because there is the next sections is held by the Sibeli. He will get some introduction about the GOAT or the EBP. So I already sent the link through the chat. Maybe if you get interested in this part, you can click the link. Yeah.
We've had to rebook it because there's been an error with Zoom.
Okay.
Yeah, so in that case, you have another 25 minutes of talking if you really want it. But I'm going to post the new link in the chat, and we're now going to start it at 4:30. So if you are interested, please come along.
Okay. So any questions? So we still have time?
Yeah, so maybe it's nice that the speakers address their questions that were in the chat, but out loud, so it's captured by the recording. So yeah, the first one was for Pablo.
Yeah.
Yeah, it's the Emilia's question about the genome annotation data.
Would you like me to address it like that?
Yeah, just
okay.
yeah.
Perfect, no worries. So Emilio was asking the following question. Are the biodiversity portals going to manage other genome annotations data apart the Ensemble rapid release? It would be nice to have an EBP genomic annotation data source where at least you can search for genomic annotations and their source, at least NCBI and Ensemble. Currently, I'm working in something like collecting all the eukaryotic genome annotations between Ensemble and NCBI, and he posted his GitHub website, and it looks like a very nice tool. So if you have seen the chat, please go ahead and check it. My answer to Emilio is that, first, thank you, Emilio, for your question. And so far, my understanding is that we are using the annotation data from Ensemble rapid release and Ensemble in general, and everything that is archived in ENA. But I don't know yet if there are plans to get annotation data from other data sources. And this is something that I will consult with my team and let Emilio know after by email or something. So yeah, thank you for your question, Emilio.
Thank you. Yeah, because by the way, it would be really interesting within the Herbal Genome Project to at least see how the main repositories, which as far as you know are NCBI and Ensemble, how they annotate these genomes. And to the best tool ever will be to then compare these two genome annotation and maybe overlap them to have an improved version of the last one, of the last two. But maybe
I
this
agree.
will be
Sounds like a good idea. Yeah.
Okay. If there is no more questions, maybe we can stop just close the workshop.
Sounds good to
Thank you
me.
so much.
Yeah, sure.
See you
First
later.
off, I'd like to say thank you to Xiaofeng for organizing the EBP segment of BGA. It has been really good and getting people aware of a variety of projects. So thank you very much.
Yeah, it's
Thank
also
you.
statement is really a lot of help
Thank you.
from you. Yeah, thank you. Thank you
And
so much.
Thank you, Sibeli.
hopefully,
Thank you
Yeah,
too.
thank you very much.
Bye.
hopefully we'll see you in the next
Bye.
session.
Thank you.